Good morning, everyone. Today is the last day of ISPC meeting 2023. Let's start our first session. Our first speaker is Archishman Raju from NCVS TIFR.
Okay, thank you. So I'll be talking today about geometric models of self-weight specification, and I'll be talking about more recent work, which is with Eric Segea and Kat Hedge-Antonakis. To set the background, development is something that for a long time has been known to be a very robust process, which you'll appreciate by the fact that you very robustly develop four fingers and a thumb. And if you think about it, that's a very non-trivial thing to be able to robustly get every time. And it's been known for a while, and in the developmental biology literature, this is known by a term called canalization. And more broadly speaking, the fact is that most of our cell types, most of our organs, they're all discrete. You don't usually see in biology half of a lung and half of a heart together. So you get these discrete cell types, you get these discrete organs, and this broadly goes under the term of canalization. Development is canalized. But on the other hand, if you think about it from the point of view of a single cell, then a single cell seems to be doing something quite complicated. A single cell has some protein, some chemical concentrations outside, and it's interpreting them in a certain way. And even the simplest decisions that's now known through years of work in developmental genetics involve several proteins. And I'm just giving one example of a signaling pathway, and you can see the number of components that are involved, even in the simplest decisions of development. So there's at heart of it, there is this kind of contradiction about how you get such a complicated process at the level of a single cell, giving such reproducible and robust outcomes at the more macroscopic level. And actually, early experimental embryology had a way to think about this, and in particular, it's given by this metaphor of C.H. Waddington, was an embryologist who came up with this idea of a landscape. And as you can see in this figure, his idea was that there could be some complicated network of interaction between genes, but they together will produce something that he called an epigenetic landscape, which is drawn here, where you could think of the cells now as sitting on top of this landscape and flowing down, and then going through these narrow valleys and reaching one of these attractors. And the different attractors could correspond, for example, to different cell types. And I would argue that actually Waddington, even philosophically speaking, was thinking of an emergent description, and in some ways this preceded our discussions of emergence in condensed matter physics. What we've been doing is trying to think about this metaphor in a more mathematical way and try and use it as a way to quantify biological data. And to do that, of course, the right mathematics is dynamical systems theory. And so you can think of development as a dynamical system. And the basic idea is simple. You can think about, for example, at the level of a single cell, the various genes that are regulated, that are produced through various interactions, whatever components you think are important for a developmental decision. And you can write down a set of differential equations. On the left-hand side will be how these gene concentrations are changing with time, and the right-hand side will be chemistry. So it'll involve production, decay, and interaction terms, which people put in various ways that they like. But the basic point to be made is that simple concepts from dynamical systems theories correspond to biological concepts. So the idea of a stable fixed point is something that you would think of as a differentiated cell type at the relevant time scales. The idea of a saddle point is something you would think of as a point where the cell is deciding whether to be one fate or the other. The domain of attraction you would think of as the region where the cell is specified to take a certain fate in the absence of any external signals which perturb it. Mathematically, the idea of a Waddington landscape is linked to something called a Morse-Smale system. There's an old idea which goes back to René Thom, but the only new contribution to this is that there's a theorem by Smale, which Thom seems not to have mentioned in his work, which says that if you have a mathematical system which is structurally stable, which roughly speaking means if you perturb it slightly, it doesn't change the qualitative behavior of the system, which is again something you expect from robustness and canalization in biology. And you have something that's bounded, which is also biologically reasonable in the sense that gene concentrations don't go to infinity. Then you can write down any differential equation describing such a system as a gradient flow, with the caveat that there has to be a metric. And so there's a pretty general way of thinking about developmental systems under the assumption that they're robust and under the assumption that they're bounded. You can think of them as gradient flows with a metric. I won't talk about the metric here, but mainly I want to keep track of the fact that you now have a potential with which you can describe the developmental dynamics. Okay, so now the next thing you want to do, of course, is you want to parameterize this potential. So you have a potential that as the cell is deciding its fate, it'll go down some potential. You want to parameterize it. You want to perturb it in different ways and see how that will change the fate of the cell. And the mathematics is kind of agnostic to how you choose to parameterize the potential biologically. You could do it with decay rates, you could do it with signaling molecules, and other ways that you have control over. But basically, as you vary parameters in the potential, you can get bifurcations, you can get qualitative changes in behavior. And it turns out only two of these bifurcations are generic. Generic in the sense that they don't require any special symmetries in the differential equations, which are the saddle node bifurcation and something called the heteroclinic flip. The saddle node, I mean, many of you are familiar with this, when here shown, for example, as a saddle point and a stable fixed point, which annihilate each other. And a heteroclinic flip is saying that here you have one saddle point and the trajectory from it goes to another saddle. As you move the positions of these saddles slightly, it changes where outgoing trajectories will go, whether to the right or to the left. And there's obviously important from the biological point of view, if you exit a certain type of cell state, you're interested in whether you go to right or left. Okay, so the final piece of mathematics before I go into the biological example is that you can now think about a two-parameter space, and you can think about three cell states, for example, three stable fixed points and two saddles, and a two-parameter space of bifurcations. And independent of the underlying differential equations, you can come up with whatever model you like. You can actually classify all of the qualitative possibilities for bifurcations under the assumption that you are only dealing with generic bifurcations. And so in a two-parameter space, you get these lines of saddle node bifurcations, and you can kind of make some arguments for how these lines can meet and write down these minimal kind of diagrams, which are all of the qualitative possibilities that exist for the dynamics under the situation that you have three kinds of stable fixed points and saddle points. And the qualitative possibilities that exist for the dynamics depend basically on the topology of how these fixed points are connected to each other. So I just want to show this slide. Obviously, there's a lot going on, which I don't have the time to talk about. So the biological example that I want to talk about is the early embryo, early mouse embryo. And in the early mouse embryo, you have a single cell, it divides, and then there are two decisions that happen. These things called blastomers become cell type A and B, they have some names. And the second decision is what I'm interested in, something called the inner cell mass becomes either the primitive endoderm or the epiblast. It turns out that biologists have some control over this decision. So you have some protein called FGF, with which as you vary that protein, you can vary how much concentration of one or the other cell type you get. So you can see how this is fitting into the framework. You have three types of cells. You have some control over it. You have some way to parameterize it. And now you can ask, how do I make a model about this? Without having to worry really about the details of how that protein is interpreted by the cell, which again is some complicated gene network. So it turns out there are two possibilities here, two topologies that you would want to think about of how one cell state can go to the other. And here, the two of them are drawn here. So in one case, you have the middle cell, A, inner cell mass, deciding whether to become left or whether to become right. In the other possibility, you have the middle cell, inner cell mass, first deciding to be not itself, and then taking a second decision whether it wants to go left and whether it goes right. And these are two distinct geometrical ways that you could think about the same picture. Okay, so now the question is, how would you distinguish between these two kinds of geometries? And our idea...
2 minutes.
Yeah, 2 plus 2, right? Okay, so our idea is that you can distinguish between these two types of geometries by doing time-dependent experiments. So in particular, if you take this geometry, I'm not showing the other one, this is one geometry where the cell first decides not to be itself, and then it takes a second decision whether to go left or right. In this, the sensitivity of the trajectory to an external control which pushes you towards the left is going to be different at different points in the trajectory. As you can see, at the beginning, if you push it to the left with some pulse of external signaling molecule, it's sort of going to follow. So this is just one trajectory with noise which happens to go to the right. In this case, you push it to the left at the beginning, but you see it comes back and it ends up going to the right again. But then if you push it later on with the same pulse, same concentration, and same time period, that same trajectory, and I've kept the same noise here just as a thought experiment, the same trajectory would go to the left. So this is, in this geometry, time-dependent perturbations to the trajectory would have a very different effect than they would on the other geometry, which I'm not, the other one I'm not showing. So finally, I'll just conclude to say that Waddington's metaphor, which I showed you, can be converted to a mathematical statement that a lot of the dynamics, gene regulatory dynamics that is relevant for developmental systems can be thought of as potential flow. Second, I've shown you that you can take models of cell fate specification in low dimensions, and you can basically classify all of the qualitative possibilities for the dynamics independent of what the underlying differential equations are. And finally, we want to argue that these potential models are actually a minimally parameterized way to fit data, and in particular, fit new kinds of experiments which can really probe the landscape at a time-dependent level. So that I'll conclude. Thank you.
Okay. Now we are open for questions.
Yeah.
You began with n coupled ODEs and then you only drew two dimensional pictures.
Yeah.
Is that obvious that it
So-
should be only solo dimensional?
Yeah, so the idea is that the phenotypical space is low dimensional. This is sort of normal in biology that you see only three kinds of cell types at that particular stage in development. And so underlying, you could have n coupled ODEs, but there's an intrinsic statement about dimensional reduction that the relevant behavior can be captured in these low dimensional pictures, which is sort of the claim about how you can fit data. But I know there's a longer conversation which I can take with you offline. Yeah.
Are memory effects not important in these systems? So you had most of the equations are Markovian, I guess.
Yeah, they're ODEs. I mean, memory effects in the sense that you could have, I guess, delay differential equations, but
Yeah, something like that.
these have been known to be important. But yeah, I mean, they tend to be more complicated. But yeah, this is for ODEs. Yeah.
Okay.
Yeah.
So how do you decide on which variables to consider to construct this fitness landscape?
Yeah. That's a good question. I mean, and that relates to the first question that was asked about what these two-dimensional pictures are. And these two-dimensional pictures we think of as sort of abstract variables, which ultimately are some projection of the higher dimensional space where the underlying dynamics occurs. But in practical terms, biologists tend to measure one or two markers. And so the assumption would be that whatever those one or two markers are, you should be able to fit all of them in this abstract space. But really speaking, it's some sort of projection of the higher dimensional space. That's, yeah.
So, in saddle node bifurcation, in subcritical bifurcation, you can have two attractors.
Yeah.
But this whole dynamics is not the result of a gradient of a potential. So, like, can that never happen in biology?
Well, so you have to, I mean, the thing is that there's a metric involved. So you can take curl terms and you can put them in the metric. And the idea is that near each of the attractors, you can think of linearizing the flow, and the flow will not be symmetric in general, right? And this is, but that non-symmetric matrix you can write as a product of two symmetric matrices. That's where the metric comes. And then you have to patch things together at the different fixed points to make it all fit. But the metric is important there to what you're saying.
Fundamentally, it's always the gradient of a potential.
Yeah, except you don't go downhill in sense of the gradient. You go with whatever the geodesic is. Yeah.
Hi.
Yeah.
So you identify the fixed point and the saddle points with some behavior in the Bechtel system.
Yeah.
Please highlight that again, and also the dynamical system do go on to chaotic orbits.
So chaotic orbits are not allowed, okay, because that's part of the Morse-Mill theorem. Chaos is not allowed, and that's also to do with structural stability. But then, what was your first question?
The fixed point and saddle points
Yeah.
are related to some behavior in the bacterial and thing that you highlighted. What they signify in your system.
So maybe this was the point, that the fixed points signify cells that are differentiated. So right in my mouse example, there were cell type A, B, and C, ICM, epiblast, and primitive endoderm. So the fixed points signify those states. Now, of course, if you keep looking in the mouse embryo, those states further differentiate into something else. And so that's why there's this thing about relevant time scales. And so when you're modeling, you have to have some sense of the relevant time scales that you're modeling. And in those relevant time scales, the fixed points signify those states. And the saddle points are really where the cell is deciding. It's, I mean, this is not something that you would capture in that kind of a biological diagram. And that's part of the point here, that those points actually are the heart of the decision-making process.
Okay, nice talk. Thank you.
Okay, let us thank our speaker.